Our first presentation is by Tom Chula from Ikarovec.
Thank you, Rishi. As a retina specialist with nearly 30 years of experience in academic roles and in leadership roles in industry, I'm excited to present Ikarovec to you today. At Ikarovec, we're building a platform of bicistronic or bifunctional gene therapy. What that means is that we have two synergistic therapeutic proteins produced from one vector. Geographic atrophy is our first indication, and what differentiates us is that we're not only focusing on preservation of retinal anatomy, but also preservation of retinal function. We've raised $11 million in seed money to date. We'll be opening up our Series A sometime this month. We've actually have verbal commitments for about a quarter of that already. We have a very experienced leadership team. Katie Binley has been working on gene therapy for 25 years, including time at Oxford Biomedica. Andrew Osborne worked at Quithera, our predecessor company, which was acquired by Astellas for its bifunctional gene therapy in glaucoma. Everybody knows Dave Geyer. Jean-Philippe Cambal is currently CEO of Vet Therapeutics, a French gene therapy company, and chair at Splice Bio, working on Stargardt disease. It just had a big Series B. We have a world-class scientific advisory board that's been very supportive, and we're focusing on large markets. This allows us to eventually get our cost of goods down and to increase our margins. At Ikarovec, we're focusing on preserving vision through two key pathways. That's neuroprotection and complement modulation. This targets the root cause of disease, and preclinically, this dual pathway approach has been shown to be superior than single pathway approaches. In addition, this has applicability to retinitis pigmentosa as a gene-agnostic therapy. Both PEDF and CD46 are important targets. They're depleted early in geographic atrophy. PEDF has been shown to be potently neuroprotective in human retinal organoid models. CD46 is a catalytic therapy. It functions at C3, which is a target of one of the approved therapies. Both are soluble, so they can diffuse through Bruch's membrane and address their particular activities at both the level of the retina and the choroid. Importantly, our combination of PEDF plus CD46 outperforms single agents and competitor approaches. As you can see here, ICAR-001 shows robust preservation of retinal structure and function. On the left, in this model of RPE loss, we can see this potent preservation of the outer retina and RPE, both on the OCT and choroidal hole mount. In the model of photoreceptor function and cellular loss on the right, we can see this robust protection of the outer nuclear layer as outlined in red in the ICAR-001 treated eye versus the null vector control. And importantly, we have functional preservation of the ERG. ICAR-001 has been shown to have safe therapeutic protein expression in a non-human primate study. We can see in red and in green, respectively, on immunohistochemistry, the elaboration of PEDF and CD46 in the outer retina and RPE. In terms of the clinical trial, it's designed to maximize success and minimize risk. In geographic atrophy, we'll be enriching for fast progressors. We'll have multiple readouts, including a 12-week biomarker readout for early value inflection. The study will basically be a 3x3 dose escalation with expansion of the two highest tolerated cohorts. We're minimizing development risk with multiple strategies, including using a validated AAV8 capsid, using an experienced AAV8 CDMO. We've gotten feedback already from the FDA that supports our development pathway, and we're currently in the process of an Australian CTN application to expedite this process. For our pipeline, ICAR-002 targets wet AMD using an office-based novel capsid. It's targeting angiogenesis and fibrosis to address long-term visual outcomes. And then ICAR-003 will target intermediate AMD using neuroprotection and complement modulation to limit progression to GA and wet AMD while preserving function. Our team has experience with multiple exits, including the sale of Quithera to Astellas. And again, Quithera was a bifunctional gene therapy platform for glaucoma. So in summary, we have a compelling Series A investment opportunity with a large addressable market. a differentiated bifunctional therapy, superiority preclinically versus competitor approaches. We have an expert team, an AAV8 experienced manufacturer, positive FDA feedback early on, an Australian CTN pathway that's underway. We've raised $11 million. We'll be opening up our Series A later this month, with about a quarter of that already committed. We have multiple milestones along the way. So if you want to be part of the solution, please reach out. Thank you.